Hello all, welcome to my channel. In this video we are going to discuss about protein, structure of a protein and function of a protein. Protein are the building block of life because they are most abundant molecules present in the body and form about 60% of a dry weight of cell. They are most common cells found in all living organisms. Apart from cells, most of the enzymes, regulatory and structural part of a body are made up of proteins. As a result, they are essential for growth and development of individual. Protein may be plant protein like seeds, pulses, and animal protein, egg white, albumin, etc. If we consider the structure of a protein, proteins are a polymeric chain of amino acid. Here is the polymeric chain of amino acid. There are two different groups, that is R group and carboxylic group, amino group or carboxylic group. So the structure of amino acid gives particular properties to protein. Amino acid are made up of the amino functional group, that is NH2. Here is the NH2 amino functional group, and carboxyl group, that is COOH. Here is the carboxylic group. Amino acids are linked together to form a polypeptide chain. One or several chain fold differently to form a protein. Depending upon the R group, here is the unique R group, there are different type of amino acid, out of which 20 are found in polypeptide chain. All these properties of amino acid decide the ultimate structure and function of a protein. The structure of a protein is classified as four levels, that is primary, secondary, quaternary and tertiary. So let's discuss one by one what is the primary protein, secondary protein, quaternary and tertiary protein. The primary structure of a protein is the linear polypeptide chain formed by amino acid in a particular sequence. Here is the primary structure of a protein, where these balls indicate the peptide chain of amino acid which are arranged in a particular sequence. Changing the position of even a single amino acid will result in a different chain and hence a different protein. If the sequence is changed, suppose this red button is at the apex and the blue button is at the base, may it change the structure and it indicate another protein, a different protein. So it is arranged in a sequence manner. Change in position indicate a different protein in primary structure. Next is secondary structure of a protein. The secondary structure of a protein is formed by hydrogen bonding in the polypeptide chain. These bonds cause the chain to fold and coil in two different conformations known as alpha helix or beta pleated or sheets. Here is the alpha helix which are coiling in shape, which have a N terminus, and another beta sheet have C terminus. This is the coiling form of alpha helix. So let's discuss the details of alpha helix. The alpha helix is like a single spiral and formed by hydrogen bonding between every fourth amino acid. Here is the hydrogen bond. Here is another one hydrogen. 1, 2, 3, 4. Fourth position there is a hydrogen. Here 1, 2, 3, 4. Here is one other hydrogen. Here is hydrogen. So every fourth amino acid there is a bond of hydrogen. The alpha helix has a coiled structure with 3.6 amino acid per turn. These are coiling structure. In our last slide we had see the coiling of alpha helix looks like this. Here is also such type of arrangement where it is in a coil form, and within this coil one turn is contain 3.6 amino acid per turn of the helix, and 5 helical turn is equal to 18 amino acids. 1, 2, 3, 4 suppose. Here is the another coiling. This 5 turn contain 8 amino acids. Helices are predominantly right-handed. Sorry, I made it left-handed, so it is right-handed coiling. As looks like in this diagram, right-handed coiling only in rare cases, such as in sequence with many glycine can left-handed alpha helices form. Only exceptional cases, that is in case of glycine, it is left-handed coiling. In the alpha helix, hydrogen bonds form between C double bond O group. Here is a C double bond O group. Here also, and N single bond H. Here is the NH group. Here is another NH group. So, in the polypeptide, the backbone that are 4 amino acids are distant. In the sequence, amino acids are distantly placed. These hydrogen bonds are primarily forces stabilizing the alpha helix. This hydrogen bond inside the helix stabilizes the alpha helix. So here is the alpha helix. Next is beta sheet. The beta plated sheet is formed by hydrogen bonding between 2 or more adjacent polypeptide chain. 2 or more polypeptide chain, adjacent polypeptide chain formed by hydrogen bond is formed the beta plated. A flattened form of a helix in 2 dimension is a common description of a beta standard or beta sheet. Rather than coil, beta standard have a bend, and these are sometimes referred as pleats, like the pleats in a cotton. These are the beta sheet here in the diagram. Beta strand can be organized to form elaborately organized structures such as sheets, barrels, and other arrangement. These are the barrel shape. Here, this is the barrel. C indicates C terminus and N is the N terminus in this photographic image. Higher order beta strands structures are sometimes called as a super secondary structure since they involve an interaction between amino acid not close in primary sequence, hence it is super secondary structure. Next is tertiary and quaternary structure. Here is the tertiary structure. The tertiary structure is the final three-dimensional shape acquired by polypeptide chain under the attractive and repulsive force of different R group of each amino acid. Tertiary form is formed due to repulsive and attractive force of a different R group. This is coiled structure and it is very necessary for protein function. P3 protein, here is the coiling structure and here is the pit acid. Next is quaternary structure. This structure is exhibited only by those proteins which have a multiple polypeptide chain. Those proteins have a multiple polypeptide chain are help in formation of a quaternary folding of a protein. So, as it is multiple polypeptide chain, quaternary molecule of a protein is a large complex structure. The individual chain are then called subunit. As there is more polypeptide chain, individual member of chain is called subunit. Here is the photographic image of a quaternary structure. This one is the quaternary structure. Next is the function of a protein. Protein is the building block, as I told. It also acts as a catalyst, transport other molecules, store other molecules, provide mechanical support, provide immunoprotection, generate movement, transmit nerve impulse, control cell growth and differentiation, as it is the building block. So here we finish with the protein. If you have any query, please comment in the comment box. Thank you.